Yeah, so thank you all for joining in to our session today. I want to welcome you to our exploration of the evolution and genomic basis of beetle diversity. So my name is Rich Adams. I'm actually filling in for Dr. Dwayne McKenna, who's actually out on the ground, boots on the ground, doing the work, collecting some samples for sequencing efforts, and I'll do my best to fill in his shoes. And joining me today in our session are four other fellow presenters. First, we'll have Dr. Terence Sylvester showcasing some of the current work in progress in beetle genomes, and also with an eye towards understanding the structure of these genomes that are being generated. Next, we'll have Dr. Nara Shin, who will be highlighting some exciting findings kind of at the beetle microbe plant interface and metabolic innovation. Then Dr. Rob Mitchell will be linking the importance of chemosensory biology to beetle genomes. Obviously, it's a fundamental life history characteristic that influences ecology, evolution, and economic impacts of these organisms. And then Dr. Kevin Moran will then showcase some nice methodological innovations that have been made to tackle some of the inherent complexities of finding genes in these newly generated genomes. And lastly, I will showcase some of our work where we are taking some of these questions to the protein structural space. So across the presentations, we'll kind of touch a little bit about community coordination, some taxon sampling, some insight gained, as well as methods used to make progress in this area. Obviously, what is really fun as we are organizing this is thinking about the vastness of the beetle phylogeny. It's amazing to think how much of that diversity is out there phylogenetically, but also at a genomic level that will likely require a lot of coordinated efforts to really understand the gene, genome, and protein level diversity that exists in nature. So as a reminder, my presenters will have about 9 to 10 minutes, and I'm going to try to leave with 1 to 2 minutes for questions at the end. But I'd love to continue these conversations with the community afterwards. Please do reach out to us, to the presenters, and so forth. So without further ado, I'd love to introduce our first speaker, Dr. Terence Sylvester. There we go. I think he just came on. So Dr. Terence Sylvester, you'll be able to share your screen when you are ready.
Okay.
Sound good?
Sounds good. Okay, let me share my screen first.
That's good.
Okay, can all see the screen and hear me well?
Excellent. We can see both.
All right. So let's go. Hi, I'm Terence Sylvester. I am a postdoctoral researcher at the University of Memphis. Today, we will explore the fascinating world of Coleoptera genomics together. Thank you so much for being here. Let's dive in. All right, so when we look at the animal diversity, nearly half of it is captured by the 4 mega-diverse insect clades. Within this, Coleoptera alone consists of nearly 400,000 named species, and it is the largest clade of the 4. Now, one might expect, given the number of species of Coleoptera, to dominate the whole genome sequencing databases, right? Well, in this figure, you can see the number of whole genome sequences available for the 4 megadiverse insect clades. The horizontal axis represents the insect orders, while the vertical axis shows the percentage of the species diversity captured by the available reference quality whole genome sequences. The values within each bar indicates the number of reference quality genomes available. So what stands out is that Coleoptera, despite being the largest megadiverse insect clade, has the least genomic representatives. Now, when we look at these beetle genomes in depth, what we immediately see is that not all beetles are equally represented. So in this figure, the horizontal axis is the beetle suborder and the vertical axis is the number of species. Colors represent observed versus expected number of sequences. What stands out here is that the suborder Adaphaga is significantly underrepresented. Furthermore, genomic data is only available for Adaphaga and Polyphaga, and there are no genomes yet for Myxophaga and Arcostomata, the 2 other beetle suborders. Well, while there is a genome for one Arcostomata species, Priagma serrata, it is a partial genome. So even at the family level, not all beetle families are equally represented. So in this graph, the vertical axis is the beetle family and the horizontal axis is the number of genomes available for each family. Out of about 200 beetle families, only 47 have representative genomes. And astonishingly, just 4 families, Chrysomelidae, Staphylinidae, Scarabidae, and Curculionidae, account for nearly 50% of these available genomes. So this uneven representation highlights the need for more comprehensive genomic studies across all beetle families. However, despite this taxonomic bias, we are now starting to see an influx of Coleoptera genomes. Here, you can see a bar graph illustrating the number of Coleoptera genomes released each year, showing a continuous growth in the genomes released. So this surge in genomic data is a promising step towards understanding the rich diversity within this insect clade. As we continue to see an influx of these Coleoptera genomes, it is also important to understand the quality of these assemblies. In this bar graph, the horizontal axis is the year, the vertical axis is the number of genomes, and the colors represent the degree of scaffolding. We see an increasing number of highly contiguous assemblies being released each year, thanks to the availability of long-read sequencing approaches such as PacBio and NanoPo and advances in high-C sequencing approaches. So far, we have about 195 chromosome scale assemblies, 101 scaffold level assemblies, and about 55 quantic level assemblies. Also, one thing we should note that there are more species with PacBio or NanoPo reads in the sequencing read archive than there are genomes, hinting there will be more contiguous genomes to come soon. Now, moving forward, deciding which species to sequence next is complicated, but it is vital for the understanding of evolution of beetle genomes. Representatives from Myxophaga and Archostomata will provide a new vantage point on the evolution of beetle genomes, and subfamily level exemplar genomes will be crucial in resolving ambiguous relationships within the order. Furthermore, chromosome scale genomes from near relatives of Coleoptera, such as Neuroptera, Strepsiptera, would provide essential information on ancestral conditions of Coleoptera, Coleopterida, and Neuropteridia clades. Now, with that, let's see what we can uncover using the available genomes. One key aspect of genome evolution is the dynamics of repetitive sequence evolution. By diving into the data from Coleoptera genomes, we aim to uncover significant patterns and insights into this fascinating process. So, to understand the repeat sequence evolution, I used family-level exemplar genomes, focusing exclusively on chromosome scale assemblies. In this figure, you can see the phylogenetic relationships of the families I analyzed, built using Busco genes. The second panel is the genome size of the exemplar species, while the third panel illustrates the repeat sequence composition as a percentage of the genome size. Broadly speaking, larger genomes exhibit a higher repetitive content, a pattern which is universally consistent. Additionally, DNA repeats and lines dominate the classified repeat sequences. So to further understand the repeat composition, I scaled the repetitive sequence to 100%, and in this figure, I removed the genome size panel. And what we see here is that in many genomes, a larger fraction of the repetitive elements are unclassified or unknown. So this highlights a significant gap in our understanding of repeat sequence evolution within Coleoptera. However, focusing our attention to the known repeat elements, we do see some interesting patterns. In this figure, I've broken down the repetitive categories into their own panels. The horizontal axis in each panel indicates the percentage of the genome occupied by each repeat category. Notably, signs are nearly absent across the Coleoptera families, and rolling circle repeats are present only in few families. And this showcases lineage specificity in repeat sequence evolution. Another way to examine the repeat sequences is by looking at the repeat landscape. And this approach provides insights into repeat composition as well as repeat sequence proliferation. So let's see what patterns we observe in Coleoptera. In this series of plots, the horizontal axis is the Kimura substitution level, or how divergent the repeat sequences are. Think of this axis as time. As the Kimura substitution level increases, you travel through time to the past. The vertical axis shows the percentage of the genome, each repeat category occupied, and colors represent the repeat category. So here, I have two genomes from the same family, Scarabidae. We see two completely different patterns here. So this peak near zero is, or zero values, is pretty common in most genomes, indicating ongoing repeat sequence proliferation or repeat sequence duplication events. There's a peak at the larger divergent values, and in these two genomes share this peak, and this could be an ancient repeat proliferation event shared by the two genomes. There is this third peak, which is uniquely present in Chrysina gloriosa genome, showcasing lineage specificity. Now, even within the same subfamily, the repetitive sequence composition is largely different. For example, here, the two genomes are in the family Cerambicinae, so subfamily Cerambicinae. However, the large genome Rosalia on top consists mostly of DNA transposons, while the smaller genome Neoclitus consists mostly LTR elements. Now, this pattern of lineage specificity persists even when we look at a finer scale at the genus level. Here you can see three species in the buprestic genus Agrilis, and all three genomes show distinct compositions and shapes of repetitive landscapes. For now, I only focused on the one aspect of genome evolution with a highly reduced dataset. Even there, we saw patterns of lineage specificity, diversity, and abundance of transposable elements. These patterns of repeat sequence evolution are just the tip of the iceberg. It leaves us with more questions about how the repetitive elements evolve across Coleoptera lineages, as well as how the beetle genomes evolve. And despite its taxonomic bias, I believe the genomes we have are a valuable asset in uncovering the evolution of beetle genome architecture and further understanding the genome basis of the evolution of beetle diversity. And with that, I conclude my presentation, and I would like to thank the Machina Lab, our collaborators, and our funding sources. And thank you so much for listening, and I would like to take any questions.
Excellent. Excellent talk, Terrence. We probably have time for maybe one question or so. I did notice one on the chat box there, Terrence, if you want to check that out. It says, do you annotate repeats in both haplotypes if those have been made available?
No, I annotated repeats only on one or the primary haplotype, because I downloaded genomes from publicly available datasets, and I focused mostly on just the primary haplotype to annotate the repetitive sequences.
Maybe one more question if anybody has. Oh, there was another one that popped up, Terrence. Asking, do you use repeat modeler?
Yes, I did use repeat modeler and repeat masker to annotate and mask repetitive sequences. Also, with repeat modeler, I generated the de novo repetitive sequences and combined it with DFAM and repbase Coleoptera specific repeats to make a library that I then use to mask or annotate the repeat sequences.
Excellent. That's wonderful. Terrence, thank you so much. I think at this point, we'll probably transition over to our next presenter, Dr. Nara Shin. But please, I saw a couple other really great questions that popped up. It would be fun to continue these conversations afterwards. These are really great. Excellent. Nara, are you ready to share your screen?
Yes, I'm
Excellent.
ready. So there's no, hold on, I will make, yeah. So can you see my screen and can you hear my voice clearly? Good, okay.
Both are great.
So I'm gonna start to talk about my things. Okay, so thank you for just being here and listening to my talk today. My name is Nara Shin, a postdoctoral fellow at the University of Memphis. And today I'm gonna talk about the convergent evolution of metabolic system in Phytopus beetles, especially Buprestidae and the Cerambicidae. So, hold on. Yes. So first, I want to give you like a quick background of my project because I don't think so many people are not used to just know about the beetles' metabolic system. So beetles are diverse in their diet and consuming almost anything. So this range from generalist to specialist feeder. And the most beetles still feed on plant and are classified as phytophagous beetles. And they spread into Coleoptera foldeni. And phytophagous beetles primarily feed on plant materials like wood and leaves. Plants are made of the plant cell wall, looks like that, and a rigid and heterologous structure. And this plant cell wall polysaccharides are highly complex. In nature, microorganisms produce enzymes that break down this structure into smaller sugar units like glucose. And these enzymes are known as plant cell wall-unit enzyme or PCWDE, and at the list up on the slide like that. And most phytophagous beetles use symbionts to help them employ this enzyme. But the group Phytophaga is different. They produce their own endogenous PCWDE without relying on any symbiont. And Phytophaga is made of two superfamily, Chrysomelidia and Coleoptera, and these Chrysomelidia have two families, Cerambicidae and Chrysomelidae. And this Phytophaga acquired their PCWD through various horizontal gene transfer events during their evolution. But today, I will focus on GH28, a common PCWD observed in the Phytophaga. And GH28 is also well-known endopolyglycinases in the microbiome to break down pectin. And most recent common ancestor Phytophaga acquired this GH28 from a fungal donor, Ascomycota, by a HG event, and many Phytophaga descendants possesses their ancestor type GH28. But interestingly, the Laminae, a subfamily of Cerambicidae, acquired new type of GH28 through another HGT event. So here's the speculated scenario. We can infer that ancestor Laminae lost their ancestor type GH28 first, but later gained a new type GH28 from Ascomycota fungi by a different HGT event. And today, most Laminae descendants possess this new type GH28 at last. And also previous studies confirm that Laminae GH28 has evolved into an orthologous group and showing both endo-PG and exo-PG activity. This suggesting functional diversification. And also we speculate that this new GH28 underwent neofunctionalization, evolving new functions through gene duplication in Laminae. But Laminae is not the only group with this new GH28. When you look at the development tree, GH28 in the Buprestidae is closely related to Laminae GH28, and which raised an interesting question. How did they acquire this enzyme? So in the Coleoptera foldeni, Buprestidae and Phytophaga, including Cerambicidae, are not close relatives, basically around 250 million years ago. And Cerambicidae and Buprestidae share a similar lifestyle. Their larvae develop inside wood and feed on woody tissue for long, extended developmental stage. So this raises important two questions. Did this GH28 enzyme originate from the same HGT event, or did they evolve in convergent evolution to adapt to similar environment? So to figure out, I employed three methods. First, I manually annotated genes from 3 Buprestidae species and 1 Tosima species using NCBI and also McKenna Lab own genome dataset, and I inferred their gene phylogeny. And I also compared the conserved gene region between Cerambicidae and Buprestidae genome. And finally, I used ARPAfold2 to compare protein structure between insect-derived GH28 and the putative fungal Ascomycota GH28. So let's talk about the GH28 in the Laminae and Buprestidae. I first started by comparing 4 Buprestidae species with 7 Laminae species. Here I marked invasive species with asterisk for your reference on the particular side. So from the manually annotated genes, I found that both Laminae and Buprestidae have different number of GH28 copies. Interestingly, this gene duplication were not related to their invasiveness, and this expansion occurred at the family or species level. And in the gene phylogeny, lamina and Buprestidae GH28 clade are not sister group anymore. Lamina has fungal sister group rather than the Buprestidae. And also from genome comparison, I did not show here, but I confirmed that there were no conserved gene reasons between these Buprestidae and Laminae. However, still we can observe both Laminae and Buprestidae GH28 genes lacks intron. So this led me to conclude that GH28 in the Buprestidae and Laminae did not originate from the same HGT event. So since they did not share the same HGT event, my second objective was to explore whether this enzyme underwent convergent evolution to adapt to similar environment or not. So to remind you the GH20 evolution, Ascomycota fungi are thought to be the donor of the GH28 to Laminae. And also fungi GH20 protein structure is well studied in many literature in the previous time. So I compared fungal protein structure to the predicted GH28 structure in one Laminae species, Anoplophora glabripenis here. So AlphaFold 2 predicted that all endo and exo PG of this Anoplophora GH28 have the same structure, and they also share the open-ended structure observed in the fungal GH28 as well. So maybe it was not that enough method to compare this protein structure, so I employed RMSD, root mean square deviation, to compare protein structure between fungal and Anoplophora derived GH28. So if protein structure has significant difference compared to reference protein, RMSD score becomes higher, like more than 1 and also 2. And this result on the table on the plot show that Anoplophora GH28 proteins have more structural difference compared to the fungal GH28. And interestingly, recently duplicated GH28 genes showed greater structural difference compared to those closer to the root. So this suggests structure change could drive neofunctionalization, although it is not always associated with it. So the remaining question here is whether Buprestidae also underwent a similar evolutionary process. So as we saw earlier, Buprestidae species have a wide range of the GH28 gene copy number, and GH28 in Agalus and Cosima are orthologous as well, and Agalus genes are embedded in the conserved microsynteny. And also the findings show that ancestor Agalus species already had at least 3 copies of GH28 due to gene duplication in family level, and most descendant species still maintain this number of gene sets. So I used AlphaFold 2 to predict this protein structure of the Agalus, and just like Anoplophora or GH28 protein in Agalus species share the same predicted protein structure as you see on this slide. So I employed RMSD again to compare their protein structure to reference one. And I found that Agalus GH28 showed significant difference compared to the fungal GH28. And also, we cannot directly infer enzymatic function due to the lack of data set, but it's likely that Agalus GH28 has evolved to adapt to specific environmental challenge and may have acquired new function. So in conclusion, Laminae and Buprestidae acquired the new type of the GH28 through the separate HGT event from the same fungal donor. And the GH28 gene in Buprestidae resulted from gene duplication in their common ancestor. And slight change in the protein structure can lead to difference in enzymatic function. So we can infer neofunctionalization based on this predicted protein structure. However, still we need more the genome data set of Buprestidae and also other Coleoptera to figure out this convergent evolution. So still this is the remaining mission for my project. So I appreciate all help and the collaboration with the lab member and current people. So thank you for listening to my talk today. So if you have any question, I'm just happy to answer them.
Excellent. Thank you, Nara. Yeah, if anybody has any questions, we have time for questions.
So if you have any question, we can just move to Rolf.
No questions, anybody? I have a quick question for you, Nara. I was just thinking about this, and maybe somebody in this audience would know. So looking at your phylogeny with the potential fungal donor, so we often think about the papers and the work showing the horizontal transfer from microbe into beetle. Is there any evidence of beetle to microbe horizontal transfers? Has that ever been documented? Has anybody? Sometimes other gene families have been known to do lots of swapping around. It's just something that popped in my head. I can't think of any, and maybe somebody else can speak to that if they know of any classical examples of this. Oh, we did get a question from Dr. Mark Blackster there. If you want to check it out, it's kind of related in the comment box there, Nara.
Okay, how much worse would the following be if the biology were indeed the result of single transfer? It can be because we could not infer that, okay, when first we could not clarify when this HET event happened, and second, maybe like a multi-HET event happened between their symbiont and their host species. So, so it could not that be worse with like a single transfer, but because only I showed the part of the GH28 phylogeny today. Yes, I think I can, but first from this Bayesian likelihood analysis, first we need to confirm that whether what is their exact or what is most the potential donor for this HT event. So before we clarify that, we could not get more like a proper or precise answer for this hypothesis. So to make this hypothesis, we need to find more, we have more like a confidence on donor species or donor class. I think so.
Well, wonderful questions. I like those.
I'll
just jump in if I can. So this is Mark. I can't type very fast. So so there's echoes here. I was just thinking that one can always postulate as many ghost lineages as you want. And so it's always possible that somewhere out there in the fungal world, there are two species extant now, which are the descendants of a mythical pair of ancestors who independently transferred genes into two different beetles. But just by Occam's razor, surely that it would be better, first of all, simply to test whether the beetle, the Coleopteran versions of these GH genes, how likely it is that they share a more recent common ancestor than they do with the other fungi out there. I think that to me would be the exciting thing to do, to actually really get in there and do some strong Bayesian tests of this, because you have a vast amount of data. It's lovely.
I think that's a great idea.
Yeah, thank you for the good feedback and good question. We also did a kind of mission because first this HT event we speculate that it happened almost like 150 million years ago. So as you mentioned that there are a lot in the gap, the knowledge between like the extinct like the fungi and something the microorganism like their evolution. So our mission is to find the answer of that, okay, where they got it and which the fungi family are the real like a donor for that. So if we can think about that, it must be have a really great like a project at all.
Excellent. Yes, that's wonderful. Well, thank you very much, Nara.
Yeah, thank you for that.
Fascinating. And great questions too. With that, let's go ahead and move on to our next speaker, Dr. Rob Mitchell. I believe you can see your screen when you're ready. Rob, you can share your screen, and the floor is yours. Rob, I do believe you're muted. Maybe that's in case you've begun.
How about now?
Excellent.
Okay.
Loud and clear.
Take two. So we'll continue this conversation and talk about the intersection between genomics and chemical ecology. There. All right. So maybe not everybody is familiar with the field of chemical ecology, but I'm sure that everybody is familiar with the idea that chemicals can carry biological information. And when we think about this, we usually think about it from the perspective of a hormone, something that carries information within the bounds of an organism. But there's really no reason we need to stop there. Chemicals can contain information externally, outside of the organism. And when chemicals do this, we refer to them as semiochemicals. So the field that concerns itself with hormones is endocrinology, but the field that studies semiochemicals and all of their varied interactions out in the world is chemical ecology. And chemical ecology is particularly important in insects because they overwhelmingly rely on smell and taste to navigate. Just some of the most commonly studied functions involve host recognition. Insects are capable of assessing and prioritizing different hosts based on their taste and smell. Insects will produce their own semiochemicals, oftentimes to attract mates to a particular location. We call these sex pheromones in that case. And the cuticle of insects is loaded with waxes, many of which are semiochemicals. It can be as simple as discriminating between males and females, or in the case of social insects, you have different semiochemicals that discriminate between the individual, the caste, the colony, the species. So there's many different functions of semiochemicals. We study them in beetles, from my perspective, from two important reasons. One, simply the sheer size of the order means that there's many different interesting evolutionary and ecological stories that we can investigate. But probably more important from the perspective of funding is that by virtue of their size, there's many species of beetles that are pests in agriculture and forestry and other areas as well. And so chemical ecology provides some really powerful tools that we can use to control species that are pests, from something as simple as using the pheromone to monitor for the presence of pests to more complicated ventures like developing repellents and inhibitors, or even trapping out populations to try to remove them from the environment. So there's a lot of interest, in chemical ecology from the pest control perspective. And classically, a lot of chemical ecology work has come in from the direction of the semiochemicals. But more recently, we've been able to look at it from the side of the chemoreceptors. And I find these especially interesting because they really define an operating space for chemical ecology. So when we think about a chemoreceptor, we're talking about a protein here that binds one or a range of different compounds. And so the chemoreceptor that activates or the combination that activates is going to represent that taste or smell in the brain. When we talk about insect chemoreceptors, that we're generally talking about three families here. It's either going to be the odorant receptors, the ORs, which are for smell, the gustatory receptors, which primarily mediate taste, and the ionotropic receptors, which have many different functions, but are involved in both smell and taste. However, to study any of these families requires large-scale sequencing efforts. So there's our intersection with genomics. Let's see why. Chemoreceptors are not small families of proteins. This tree right here shows us the gustatory receptors, just the gustatory receptors, from a single species of beetle. So these are enormously large radiations. The genes are highly divergent, which means that we're not going to be able to go in easily with primers or universal primers or anything like this. We need genome scale sequencing. Things get a little more complicated because chemoreceptors tend to be formed of multiple small exons with low homology. And these tend to be smeared across, 100 kb of genome space more often than I would like, which makes prediction of the small exons especially difficult, not aided by the fact that there is somewhat creative splicing when we're dealing with chemoreceptors. I couldn't tell you why, but many times what we expect for splice sites tend to change quite dramatically in chemoreceptors, which also frustrates splice site-based predictive methods. At this point, you're probably wondering, why do we even bother with the genome? Let's just look at the RNA. but we run into problems there. There's very uneven expression in chemoreceptors. Yes, you can capture full-length models from the RNA, but you're going to get an equal number of partial or fragmentary models that are difficult to use, and you don't know what you're missing. From my experience, usually less than 50% of the chemoreceptor repertoire is going to be expressed even with deep sequencing of enriched tissues like the antennae. So you're going to miss a lot if you focus only on the RNA. All right, so what are you supposed to do? What do we do with this information? Well, yeah, you could ask for a collaborator's help, but I'm going to argue that it is possible for anybody to create a high quality annotated set of chemoreceptors if you follow some general principles. First, you have to have that high quality genome, and you need to supplement this with tissue specific transcriptomes from the antennae or the palps, which will really help for those small exons. You have to assume that there will be manual annotation. So you can't rely solely on prediction, which means you do need a good local database of high quality gene models, ideally from the family that you are working in or a related family, but at a minimum, it needs to be from the same order as you're working in, because once you breach that ordinal level, the homology between chemoreceptors drops dramatically. And so any homology driven search like BLAST or DIAMOND is going to be very low efficiency. Finally, we can take advantage of the radiations of chemoreceptors. So it's most commonly found, or at least in my experience, that a given gene is going to have at least one paralog, usually more than one. And so as you recover these gene models, you can do iterative blasts, continue to blast with the new models of genes, and very effectively and quickly build out the locations and the identities of chemoreceptors that you missed in the previous part of the annotation. If you're interested, manual annotation is getting easier all the time. The predictive models are getting better. So our following talk is going to focus on some new approaches to this, some very effective approaches that have been building out these large multi-gene families, specifically the ORs. So Kevin will throw some big data at you in a couple more minutes here. Applying this, right, we need to have a lot of genomes. We need to have a lot of transcriptomes. And so I am part of a larger collaboration that's looking at the phylogeny of longhorn beetles. In the process, we're generating a lot of genome information. So we're piggybacking on this for the chemoreceptors. From the cerambicids themselves, we're pulling genome from all 8 of the subfamilies. And then we're also building in genomes from the allied families of beetles. Layering onto that, we're doing replicated antennal transcriptomes from 3 males and 3 females from each species. And on top of that, another 35 to 40 species, depending on the material that we can obtain, to get additional antennal transcriptomes. And so our goal is to really dig into this and try to get good, solid chemoreceptor models, at least for one family of beetles. This is ongoing, but we have a little bit that's come out already, focused on the red milkweed beetle. And this is going to be the focus of our session chair, Rich's talk in just a moment. So I don't want to get into too much of this, but I will point out that our methods generated about 100 ORs and GRs. And most importantly, these came out with a really high degree of confidence. So we got almost all of these ORs within and GRs within 95% of their predicted length, and most of them were full length. The comparative work has also been very beneficial. So we're seeing all kinds of interesting things now with apparent functional groupings in the chemoreceptors. For instance, here, those blue ones, you can see that there's an ortholog from each of the species that's in there. We've got really short branch length. So there's some sort of functional grouping that's starting to come out here. I'm really hoping we can get into that. But actually, at the moment, I'm much more interested in another nice side benefit of the transcriptomes, which is the expression data. And so we can pick out these chemoreceptors that are really highly expressed and go after them. And we're starting to see that these are associated with pheromone sensitivity. And so this is pushing us towards some really neat stuff with pheromone receptors now as well. But unfortunately, I am getting towards the end of this talk. So I'll leave it to Kevin to keep going on about chemoreceptors. I want to make sure that I thank everybody who's been involved with this very large scale project. And if there's time, I can take a few questions.
Wonderful. Thank you, Rob. That was great. Yes, we do have time for questions, so please ask away, y'all.
Can I just ask, Oh, God, sorry, we've got echo here again. Can you predict what the receptor will bind using AlphaFold and docking?
Yeah, that's a good question. There's been a lot of interest in this for clear reasons. There are some associations when you do docking simulations between what looks likely to bind, but really the work needs to be done in an expression system to get good data. And so it needs to be in the most common ones are phagocytes or human embryonic kidney cells. There's what's called the empty neuron system in Drosophila, where you can put in a gene and then measure the sensilla activity. But you can get some hints from folding and docking, but I wouldn't rely on that for anything beyond maybe suggesting targets for the expression systems.
It worries me if we're getting, let's just pretend there's only a couple of hundred of these per beetle genome, and we've already got 200 beetle genomes. I'm already at 40,000 in my head, and we're going to be at 1,000 beetle genomes soon. So it's going to be 200,000 receptors that we need to know what they bind, so that when we put the pheromone out in the field, we don't catch all the wrong beetles or catch all the wrong males when we're trying to get females or whatever. So is it not, do we not have to do that? Sorry.
Well, I'm not the right person to ask. I'm not really on the leading front for modeling and folding. And actually, Rich has been doing a lot of this stuff. So maybe, Rich, you want to chime in? Have you seen any interesting stuff with the folding work?
Well, I think that's a really great question. There's still a lot unknown. I think that maybe it can open some doors. There's been some interesting papers that folks have used for using AlphaFold 3 to also find potential ORs and things. I think it definitely is an area of interest. Maybe it would be great to find systems to also experimentally validate what is being done on the computer. There's like a lot of opportunity there, but I think you do pose a really good question. What are the numbers needed? They're very funny proteins because there's like a level of conservation, but also it only extends so much. And so like Rob was kind of lamenting over these kind of almost like there's conservation, but you need something relatively close. But the homology of some of the exons starts falling off, but it's very interesting. But yeah, really good questions.
And it's not a problem that's restricted to the beetles. I mean, we have hundreds of chemoreceptors from every insect species that's out there, and we want to know what they all do. And when we're bottlenecked by that functional work, it's slow going. And there are very few functionally characterized genes in the beetles and really broadly across the insects. So it is a problem.
Yeah, the question was just stimulated because I read an Lepidopteran OR paper this morning, was just published in GBE, which just blew my mind as to how many they have as antennal ORs. So,
well, that is a wonderful segue actually to our next speaker. Kevin Moran actually has been working on some developments for this and has some promising results. Kevin, are you on with us?
Yes.
If
Hello.
you can, maybe start sharing your screen and I can, okay, I see you over there. Awesome.
Am I sharing now?
Excellent, Kevin. Yes, you're good to go.
Okay.
The floor is yours.
Get this started. Everyone can hear me?
Yes, we can all hear
Okay,
you.
great. Let's minimize this. Stick that up there. So, hello, my name is Kevin Moran. I'm a postdoc in the McKenna Laboratory at the University of Memphis, and I'm here today to talk to you about methodological advancements in odorant receptor finding and annotation. So, for people that may not have been here for the last talk, insects detect odors using odorant receptors, which we're going to shorten to ORs. And ORs work by matching the shape of odor molecules. So within Coleoptera, Mitchell et al. 2019 classified beetle ORs into 9 subfamilies, and he did this using 1,200 ORs from 10 beetles and 1 strepsipteran. So the problem here is that he did this. Difficult part here was that he had to manually curate the reference set and manually curate the set and correct them and stuff like that. So why is this not moving? There. So I'd like to say that OR annotation and finding is a problem in search of a solution because ORs are among the largest gene families in insects' genome, but they require expert manual annotation for accuracy, but this is very slow. Automation has struggled with this clade, and in part this is due to the tendency of ORs to be arranged in tandem arrays. So as you've seen from previous talks, the availability of new genomes is exponentially growing. So clearly we're in need of a new solution beyond just manual annotation. So as part of my PhD and continuing to my postdoc, I've been working on a phylogenetic pipeline, which I've expanded into other skill sets. So I like to say you have a genome, but no annotation. Is patience not among your virtues? No problem. We're just going to map the exons directly to the target loci from the assembled genomes. So behind the curtain, the way this pipeline is doing things is it takes the genomes and it's going to artificially convert it to shorter sections. We're going to chomp it into shorter overlapping sections to create a tiled read all the way through the genome. Then we take those, we map them using Diamond to our region of interest. We identify the clusters. We run that clustered area, that whole cluster through HMR again to catch anything Diamond might have missed. And only then do we align these exons to the references and then splice them together. So I started with my work with phylogenetics, and I'd always see all these genomes, lovely genomes out there that hadn't quite been annotated yet, and I'd want to pull them into my data set. So I developed something to go out and grab the genome and just pull out all the USCOs and splice them together. And it was one day that my PI said, "Hey, do you think this would work for multi-copy genes?" And internally I said, "Uh-oh." But outwardly I said, "Well, let me give it a shot." And 6 months later, I said, "Yeah, we can do it." It took some work, but it works. So here you can see this is a multi-copy OR, and it's mapped against the reference here, and you can see each of these is the exon as it exists in the genome, which have already been properly spliced. So this technique was quite accurate. So what we did is I did a blind comparison using Tetropes tetrapithalamus, and the pipeline predicted 127 OR clusters versus 121 by Rob, the expert Rob. And this difference was largely driven by pseudogenes. So my pipeline has a little more wiggle room on what it considered a pseudogene. Regarding the exons themselves, it was about 90% accurate. It recovered about 90% of the coding exons. The exons it missed were short and or divergent. Sometimes these exons are short as a single nucleotide, and obviously a mapping approach is not going to catch that. I will also say that recovery decreased with phylogenetic distance, and it could be as much as including a family level exemplar in the reference set could boost recovery as much as 15 to 30 exon recovery by as much as 15 to 30%. And splicing was about 90% accurate. As Rob said, there's a lot of oddballs and stuff like that in the ORs. So hard to find a one-size-fit-all approach. So having had this, what did I do with it? Well, I decided, let's strip mine all the Coleoptera genomes and see what we get. So I strip mined the 350 genomes across 45 families, which left me with 43,000 putative odorant receptors. And the nice thing was that with the pipeline, this took less than a day. We don't have to do anything like repeat masking or anything like that. We just rip the exons right out, which left us with this tree, which we're going to reduce. So interestingly, while it's still a preliminary tree, it's not the final tree, we do get a different tree from Mitchell et al. 2019, in that while it covered most of the structure he did, it splits group one, with this group one clade up here consisting of the mixophagans, the archostomata, and the autophagans, and early diverging polyphaga, while the second group one is late diverging, more derived polyphaga. So we can also use this data to look at the presence of the ORs across Coleoptera. And there's some interesting takeaways here. For instance, the subfamily 2A is present all across Coleoptera. We can also find that subfamily 5A evolved within polyphaga, and that loss of subfamily 4 appears highly correlated with 5A, as we see multiple independent losses of 4 after the evolution of 5A, while as beforehand it's rather rare for 4 to be lost. Interestingly, we also find that some families of Tenebrioidea do possess all OR subfamilies. The other thing we could look at is expansions of these OR families across Coleoptera, and it's quite an interesting quite some interesting statistics here. For example, OR subfamily 1 has a massive expansion in the scarabs, but not in the related not in the other Scaraboidea. While in the we see a massive expansion of 2A in Tenebrionidae, which is linked with the massive expansion of 5A. In the Elaterids, we see a massive expansion of 2B, and 3, on the other hand, is the most likely it has the most independent expansions, appears to have the most independent super radiations of the subfamily. 5B, on the other hand, is the only subfamily of ORs which has no super radiation across Coleoptera. And 7, interestingly, is relatively stable across Coleoptera until we get to the Phytophaga, where especially in the weevils, it experiences a super radiation. So someone just said that they were impressed by how many ORs that the Lepidoptera have, and I'm wondering how does that compare to some of these beetles? Because the pipeline predicts some of these beetles have an enormous amount of ORs. For instance, Zophobus morio here, it predicted 1,009 ORs, which is an enormous amount, and I would not envy the person that has to check all those. But you'll also see here that the Tenebrioidea typically have a super radiation of a very high number of ORs, and specifically the family Tenebrionidae. So to take away from this example, I was able to achieve the goal of providing rapid identification of ORs. These identifications had utility, as you were able to see. We were able to produce a phylogeny. We were able to look at their expansions and their presence. I will say, however, that full annotation remains problematic and still requires expert review. However, it provides a strong starting point for experts to start with. Rob estimates he could cut easily a month or more off of each genome he worked with. But I will say the greatest remaining challenge is the identification of these short exons, and it will require a technique beyond mapping to recover these shorter exons. And finally, I'll end with saying that this technique I presented here just on the ORs, I have tested across a broad range of other gene families, such as many of the GHs and other compounds related. And finally, I wanted to thank the other members of my PI, Dr. McKenna, other members of my lab, and our collaborators. And with that, I would be able to open it up to questions.
Excellent. Thank you, Kevin. In an effort out of time, I might go ahead and jump in and encourage folks to, I already noticed several really great questions in the chat section that are popping up. If you want to give maybe pick, maybe if you don't mind, Kevin, answering those comments while I get going, and I will keep my talk very short so I don't bleed into the next section.
The pipeline is not available yet. It's a multi-year effort, but I'm looking to have it published within the next 6 months. So soon. I have not tried it with tandem gene arrays. It has no problem with tandem gene arrays because when we chomp the genome, we keep track of where the chomp sequence originated from. So we're basically we know the location at all times, and so but it also keeps track of where it is on the reference gene, so it won't allow it to go. That way, you can basically know if we go back to where you are in the gene, so it resolves the tandem arrays that way by knowing where it is in the genome and where it should be on the gene.
Sounds great. Thank you so much, Kevin. Yes, please. I love these conversations. Please continue these in the chat. while I get going to try to wrap up, and I promise I will keep mine short and sweet, hopefully. Excellent. Let me see real quick. I can start. Excellent, y'all. Well, that was wonderful presentations. To kind of follow up about these questions, how do we tackle some of the analytical challenges that are facing the megadiversity of beetles and beetle genomes, beetle genes, beetle proteins, and all of that? And so I want to showcase a fun little project we've started that is kind of taking these questions to the protein space. I want to introduce you to perhaps my favorite beetle. I know it's hard to say that, but this one's pretty close. This is Tetropes. Y'all might know it as red milkweed beetle. So this one right here is actually taken just outside my office. This is the common red milkweed longhorn beetle, Tetropes tetrophthalmus. And Tetropes feeds on milkweeds in the genus Asclepias. So together, these two organisms have been the subject of well over a century and a half of really amazing research into the interactions among species, into the evolutionary history, the evidence that they have presenting a really curious case of codivergence between plant and insect. There's just a really rich body of work involved in this system. And so recently we sequenced the genome of Tetropes tetrophthalmus, and I will provide a link at the end where you can take. Good thing about taking presentations online is I was trimming things as I was going, so I took a little bit of that out. But we sequenced that genome. We looked at all sorts of different things. We looked at some of the metabolic enzymes that Nara talked about. We looked at the olfactory genes that have been discussed. And of course, here's a picture of Tetropes, almost like in the 1950 horror films of its coming to crash through the city. So you can really appreciate the exaggerated antennae of these organisms. And so in our analysis of the paper, with a lot of Rob's help, we identified all these olfactory genes. Some of them were complete, some of them were incomplete. We had about 121 total, including some incomplete models. And so given the nature of beetle genomics being these non-model organisms, right, where the biochemistry may be a ways off, and we wanted to see, hey, can we leverage some of these new analytical advances in deep learning to learn something about the protein diversity of olfactory genes? So what we did was we took all of these OR genes, all 121, and we fed them into Google DeepMind, this robot right here, just into their algorithm through the Google Colab Network notebook. And for each of these, we essentially received a predicted protein structure, okay? We actually, in this analysis, we used a couple of different algorithms. We compared them, contrasted them to see, hey, is there evidence that one algorithm will do better than another? I won't show you some of those results today out of time. But what we were allowed to do is we looked like, okay, cool. So we have this beautiful OR phylogeny for Tetropes. And here's what we can look at. We can visualize this is the high confidence model from each OR group, one from each of these, including we also have an orco predicted structure that we could compare across species. And so next we set out, how do we explore these like analytically on a quantitative level? How do we explore the diversity of these proteins? So we set out on a journey, and we took each of these OR proteins and we computed the structural distance. We tried a couple different distances in this work. Most of them are based off a root mean squared distance, similar to what was mentioned in Nara's talk. So what this looked like is you compute all pairwise distances, well, except for ones with itself, because we know those were 0, between this OR and this OR, this OR and this OR, and you can imagine what this looked like. I want to mention a graduate student who was working on this, Matea Duncan, as well as an undergrad. They learned how to write for loops to automate. We got really good at writing for loops to automate these analyses. So we took all these basically this giant matrix of pairwise protein structure distance. We fed them into an unsupervised machine learning algorithm. Y'all may be familiar with this one, T-distributed stochastic neighbor embedding. That's the mouthful, or TSNE. Those of you who use single cell RNA data or tissue level stuff, this is very common to separate out. It's sort of akin to the ideas of what we use when we use PCA and so forth. And so we projected on the left, on panel A, we projected those pairwise protein distances into this multidimensional space, looking at the first two coordinates. The asterisk or the star there is from AgLab, the Asian longhorn beetle, and the diamonds are Totropes, and the colors represent the different OR groups. On the right, we use the sequence-based evolutionary distance from the phylogeny. So a couple of things we noticed. One, but even the structural distances do recapitulate our expert level identity of these ORs that was recovered with our collaborator, Rob Mitchell. So that was a pretty good job. You can see a lot of clusters. But we also see some interesting patterns within a certain groups, right? So some of these groups we actually saw, like, by the way, this is Orco right here, but we saw sort of a divergence of several different orthologs that are kind of separated in their structural space. And kind of it's kind of similar here in group 7. And then the largest divergence between sort of axis of variance coming from our group 7 as well as our group 1 and group 5A. And so we also kind of explored this using the sequence-based evolutionary distance to see, hey, how well do the two mirror each other? So one thing we tried was, hey, let's take all these pairwise protein distances. Within a species, red is Totropes, and then blue is Asian longhorn beetle. And then these triangles are between species comparisons of proteins. And we then modeled protein distance, root mean squared distance as a function of sequence-based or evolutionary distance to see, hey, what type of patterns. We do find evidence of a positive association between these two, which makes sense. I do want to draw your attention to one particular area of this plot. Over here on the left, these proteins sort of caught my eye as we were running these analyses, because they represent a set of olfactory genes with fairly high protein level distance, but relatively lower sequence-based distance, which is kind of interesting. We know that the way molecular evolution acts on amino acid substitutions, they're not all the same, one and the same, different. Sometimes one amino acid substitution could have a much bigger impact than a couple, depending on the type of residues that are being swapped. And so we're like, what are these interesting proteins? And right now we're kind of exploring those. A lot of them, here's just a sample of those genes up, those ORs up in that upper left corner there, are coming from our family, our 5A and 5B groups, but a couple in 2B and elsewise. And these are a couple of those proteins here that we're kind of exploring, trying to understand what is the structural and potentially down the road, the functional impact of even relatively few substitutions and what that may mean on olfactory biology. And we have a lot of plans to explore these types of analyses in detail. This stuff is pretty hot off the press. Maybe understand which parts of the protein are most relevant to these changes. And with that, I'd love to thank my lab. I'm a relatively new assistant professor at the University of Arkansas. These are some of the folks in my lab who are working on this project in its varied dimensions, including my graduate students, Matea, Jennifer, undergrads, Emma, Emily, and Jack. And I'd also love to thank you for listening to tuning into our session. I appreciate all of our collaborators, specifically the McKenna Lab and the Mitchell Lab. And thank you all very much for listening in. I think I'm just over my time allotment. So with that, I would love to hand it, I think with the time, hand it over to our next committee. Would make the most sense for the I5K community meeting itself. So thank you very much.
Yes, thank you, Rich. This was an amazing set of talks in these previous two sessions. So let's thank all of the speakers from both previous sessions. And, it's a good foundation for the I5K meeting, which is coming up now. So if you want to go, please do. But we're going to have updates now on the new exciting directions that the I5K organizations has taken which help sort of help drive the community behind the amazing science that we've seen here. So please stay tuned for this, and We're also going to have some discussion at the end with the breakout groups that you're all welcome to contribute to that will help us shape the direction of I5K. So without further ado, Brad, please take it away.
Yes, hopefully everybody's seeing a screen directly in front of them with our community meeting that's going to get started up now. Just a little bit past the top of the hour, but some great discussions beforehand. So appreciate everybody's input prior to. So obviously we have a QR code here. I was hoping to have this popped up for just a few minutes for people to quickly grab out their cell phones and scan. It's a link to various links that'll give you information on the I5K. Again, I'll peruse that a little bit of information as we go through these slides. So give just another 2 or 3 seconds.
Brad, you're muted.
Like a Zoom rookie here. So I was giving a little bit extra time on that first slide, just so people can kind of give a little bit extra time and to go to that QR code. So hopefully some people went about doing so. So kind of just kicking back in the I5K initiative, who exactly are we? We are a global presence, we have global participation, and moreover, we're a community. And we're a set of resources, and overall, we are basically collection of everybody. We are you. So it's got a little bit of an indication of our resources that we have available. We'll be going over these a little bit more detail as the presentation goes forward. We have various resources that have been put together as far as workshops, seminars, symposia, as well as different things on social media. So the collections of resources that we have are defined by our vision. We are, just verbatim, we are members of the global genomic community to ensure that all insect and other arthropod scientists have access to reference quality genomes and insect and arthropod genomes, and the information needed to perform the highest quality science with their genomes. So kind of, again, reiterating, a lot of contributing information and knowledge is built to bolster this community and generate greater impacts just above and beyond your individual work. You know, we're a collective. Train current and our future scientists kind of build up the information and the infrastructure for the scientific community as a whole. So it kind of leads into just who exactly are we within the I5K. We're bridge everything from individual scale projects to larger initiatives such as we just heard about, the BNL 100, Ag 100 Pest. We're various institutions, universities, United States Department of Agricultural Research Service, various data repositories, data catalogs, including I5K workspace, which I know probably is running, a co-runner of, and we'll be talking about a little bit later. We're various museum specimens. We are working with a lot of the technology companies out there to develop those small input library methodologies, as well as we're different methodologies that we're kind of developing pipelines for sample collections, and analyzing deep evolutionary times, and so forth. So all this is kind of defined by our mission. And we kind of did a revamp, what we called it, late 2023 and on to early 2024, kind of reframe I5K in more of a post-genomic context. And one is to promote the best practices for genome projects, how to best approach them, how to get the most out of your data, how to design your experiments appropriately. That's always very important, obviously. And we want to kind of transition the genomics into the post-genomics area and provide a lot of support for the generation access and various other aspects of genomics to help the community move forward. And also present some recommendations to fund holders. So we're acting not just as individual groups, but to provide bolstering or foundational support to grant submitters, that there is a group of pipelines of methodologies, and something that will help ensure the review panel that this will come to fruition. So again, within that revamp, we divided ourselves somewhat into working groups that would help kind of put forward that mission and our vision. And this is all directed under the coordinating committee. But underneath them are a lot of other folks working really hard to kind of keep things moving forward. And I'm going to cover the community building and outreach, as well as the executive committee. So kind of some of the things that are done within the context of those working groups is listed here, but I'm going to go into much greater detail working forward. So the executive committee, every group needs a one that defines the bylaws, the outlines how things are going to proceed, what things are going to move forward in what order. So this group kind of guides the general direction and the governance, develops our agendas for the meetings, identifies the priorities, coordinates various interactions amongst the working groups, how can we move forward in the best and meaningful way possible. Put together various meeting presentations for access points of contact, and of course, right now doing some recruitment, hopefully. So then the last thing I will be covering is the I-5K community building and outreach. Seems to be quite the active group. This one puts together all the conferences, symposia. Some of you hopefully have been attending or heard of the Arthropod Genomics Symposium, as well as the AGSX Arthropod Genomics Symposium X. That's been going on virtually for the past 4 years, and it'll be coming up in 2025 as well. There's symposia at the ESA, the Entomological Society of America, Plant Animal Genomics, and of course here at the Biodiversity Genomics for at least the last 2 years. And we coordinate the social media outlets. Hopefully some of this information for this particular meeting went out through and you're received through those social media means. And the YouTube channel in particular, again, with that link tree giving you access to all those social media links. Post a lot of videos and trainings that are very helpful and educational to the community. I don't have the stats up here, but hopefully that'll come in a subsequent slide. And I think this is where Monica takes over.
Yes, thanks, Brad. Hopefully everyone can hear me. I'm Monica Polschow. I'm also, along with Brad and a couple other people who I see here in the room, part of the I-5K coordinating committee. So we're here to represent the whole community. So I'm going to take over with our remaining working groups and then some talk about our discussion for today. So one of our active working groups right now is the training working group, where one of the needs that we identified within the I-5K community, which it seems to be pretty generic across the board with many people doing genomics, is the need for training for particular fields within genomics. And there's a lot going on in that space now, I'm sure, within this seminar or the whole BG 2024 series. We're going to hear some of those needs too. And we had the BGA Academy prior to this with many wonderful tutorials on how to do things in genomics, but this is also a problem in the insect realm. And so this working group wanted to identify specifically within insect genomics, what are needs for training and what can this group help provide with the resources that we have. And so they focused first on Apollo, which is a manual curation tool that we host at the I-5K workspace, which I lead. It's a database for data discovery and insect curation. And so they put together tutorials that where they deliberately made them small, bite-sized, so that if you have a particular question about what to do, you have a 5-minute or give or take YouTube video with the transcript that will help allow you to figure out that question. So they're working on those, and those are going to go up on the YouTube channel that Brad just mentioned soon. And so stay tuned for those. And also, this group is actively looking for feedback on what sorts of insect-specific or in general training needs that the community has for future work. So please reach out to us if you have any ideas there, or if you'd like to join this group and contribute some training. Next slide, please, Brad. Okay, next I'm going to talk about our other active working group, which is the I-5K standards working group. So this one came about when we surveyed the community, similar to what we're going to do later today, to ask about what the needs are. And the community clearly requested guidance on best practices and standards for genome projects. A lot of these are currently being developed by many organizations under the EBP, Earth Biogenome Project umbrella. For example, the Darwin Tree of Life, we've heard a lot of Mark Blackster today. And so that group has been really key in driving a lot of the standard setting. But we were curious and wanted to see if within the insect or arthropod specific space, if there were gaps that were being left by these larger projects that we could help articulate and identify and then raise within the larger community. And I5K, as an established organization, we're already in an ideal position to advocate for standards, including those that are already developed by these other organizations with all the hard work that they've done. So our goal within this working group is to provide specific, useful, and up-to-date guidance for the I5K community and beyond regarding standards for arthropod genome projects. Can you hit next, Brad? So what we did last fall, we convened a group of experts to perform essentially a gap analysis on the current schematic of the genome project workflow. So what I'm showing here on the right-hand side of the slide is the very high-level workflow that any group must go through if they are to complete the sequencing and assembly and annotation of a genome. This is derived from a Earth Biogenome Project paper that you can see the citation for there. And so we broke out into groups to discuss really what are still problems that we can identify with these gaps that need articulating and solving. Next slide, please. I'm not going to show you what the answer is. We're working on the manuscript right now. So these are our next steps where we're describing key gaps as identified in this workflow. But stay tuned. We have some interesting insights and things that can be worked on in the future that dovetail with existing work that's already ongoing. If you want to see them, Mark Halfon from our working group actually gave a longer talk on this during the BGA series, which I think recording should be available for that. We also want to, so within this manuscript, we're going to describe those key gaps, but also provide these best practices recommendations of existing standards for writing, and then also for submitting genome reports also. So going not just into the data generation, but also into the publication scheme of things. For the long term, we are hoping to develop educational materials to advocate for or disseminate information about existing standards. So in our analysis, we distinguish between gap types, and one of these is a practices gap. And so this would help with solving some of those. And we also need to form teams to help develop needed standards. So if you are curious about joining us and participating in the future, there will be opportunities to join new working groups on standards development for those gaps that we identified that need some closing immediately. Next slide, please. So our final working group, which we're also very much actively looking for people to contribute to, is genome project coordination. So the idea here is that within I5K, we're a larger distributed organization where essentially we can all be part of this larger group. And we've heard articulated the need to be able to understand who is sequencing what at what time, right? I want to know if a certain species is being sequenced so that my graduate student who may need the same genome doesn't put a lot of work in and get scooped or resources are duplicated and that sort of thing. And we can see a lot of what's already ongoing in that space already through the Genomes on a Tree portal, which I'm showing the link for here, which was also spearheaded by the Darwin Tree of Life project. I think Mark can correct me if I'm wrong there, and has lots of really great information right now that you can see about genomes that are not just completed in NCBI, but are ongoing and in progress that really help inform this sort of planning aspect. And so where I5K can really help out and make a difference is if we galvanize the community to help start reporting that sort of information in advance. So I'm showing here some examples from the Genome 100 project that we heard about in the first session with some great information here. So on the left, you're seeing a graphic on counts of numbers of species that are in different categories. And if you look at the very left column, that's the number that are still in the data generation stage, which you probably wouldn't know about if you just looked at what was already completed in NCBI. Right. So this gives you some good information. And then if you can click on and further identify on the right what species are being sequenced and lots of good information about those sequences as well. I can't see, I'm not showing right now all the different types of information, but there's lots of good stuff going on there. And so the idea of this working group is to be able to advertise this resource to the larger community and advocate for using it and provide tools to actually interact with it that supplement the great tools that this group has already provided to help out with this project. Next slide. All right, we'd be remiss to not acknowledge all the amazing people who have been doing work on these topics. So here's a list of who's there, and then you can just see your name on the slide in our next talk when we give this, if you'd like to join us. And the QR code that Brad had before would help us give us information, and we're going to give you information about how to reach out to us. And we're going to show that QR code later on, I think, too. All right, next slide. Okay, we need to also sort of tout our accomplishments a bit. So we've shown what we've done and how we're doing it. And this has led to, through those working groups, but also through our past 12-year or so history, we've amassed some accomplishments and metrics that we're showing right here. So I5K itself is totally volunteer-based. We're unfunded, but we've been able to inspire funding essentially for some other projects. You see a couple of these here. The I5K pilot in 2012 or 2013, and since then a couple other projects, two of which we heard about in one of the previous sessions, Ag 100 Pest and Genome 100. And so 171 genomes have been released since then, with more, as Raina said, obviously more are going to come out soon, more coming. The I5K workspace at NAL is a database for data discovery and gene curation that we mentioned before. And that's also a project that was inspired by I5K and came about because of this community effort. As Brad mentioned, we've been very, very active with outreach. So the Slack community that I think Rob Waterhouse initially started with over 2,000 members, where you can get lots of great information about genomic techniques or ask questions and learn about webinars like these in the future. The YouTube channel with over 13,000 views that Brad mentioned, and the AGSX webinar series that the outreach working group and others organize also. And we have a couple of flagship publications that we'd encourage you to check out as well, listed down there. Okay, next slide. All right, so that was our whirlwind tour of the history of I5K and where we've gone in the past couple years, sort of in light of the many high quality new genomes that are coming out thanks to new genome assemblies technologies, but also all the community efforts behind getting these genomes out and understanding the priorities. So what we wanted to do now, I mentioned and Brad mentioned before that we're really keen on feedback, and that we actually actively integrate user and community feedback into how I5K operates. That's what we do. I5K is very much about community. And so what we're doing now is part of this data gathering. So if you'd like to continue on with the discussion, please stay along. We're going to talk about two topics, sort of what the barriers are for you for performing insect genomics. And the second would be what would help pull more people into the active fold of I5K. So next slide, please, Brad. So here, we're going to do this discussion session via breakout rooms. So we have our wonderful host of the meeting, Glenn Haynes here, who's going to generate 5 breakout rooms, and we have 5 co-hosts here who will also be moderators who are hopefully already made co-hosts in Zoom. So Brad Coates, myself, Jay Evans, who you heard from earlier, David Molick, and Josh Benoit, who are also very active members of the I5K community, will be your moderators for those sessions. So they will join your group, moderate questions, and record your thoughts. Then we'll regroup, come back together here 5 minutes before the top of the hour for a final summary and next steps. So just general code of conduct. We don't have an explicit one out here, but it's essentially be respectful of all participants and encourage discussion of everyone. We really welcome everyone's contributions here. And thank you for your participation. So next slide, Brad. These are the questions that we'd like to discuss. And so we have each of your moderators should have those questions at the ready too. So once we go into breakout rooms, you should be able to see it. And the 2 questions are, as I mentioned before, what, if anything, is preventing you from creating new arthropod genomes or working with them, or working with existing arthropod genomes? And the second question is, if you are not yet active in the I5K organization, what would motivate you to contribute? So thanks. And so Glenn, if you can start up the breakout rooms now, and get us into those and we can start discussing.
Moments away.
Great.
In
thank
fact,
you.
I'll start all the
Excellent.
rooms and then you'll be able, I'm still adding the names of the people. All the rooms are separate, of course. If you have a problem, say leave, go back to the main room, and then I can adjust you however we need to be.
Great. Thank you. Yeah.
I've
And
opened
then,
the rooms.
great. Josh, David, Jay, Brad, myself, we all know our numbers. So you find your room and join. And everyone else should be assigned randomly to those rooms. So hopefully we should be able to do that. If you don't know your number, let us know now. So, David, you're 1, Brad, you're 2, I'm 3, Jay, you're 4, Josh, you're 5. So,
I still
right.
need to rename 2 rooms. Okay.
All right. See you all on the other side. Thank you.
I take it,
Room
everybody.
4 is Jay
Oh,
Evans.
I guess we don't have those. So here we go. Well, thanks to everyone here. I don't want to take up any more of your time since I think we're at the top of the hour. So I appreciate everyone's contributions. Here's again is that QR code if you missed that before, if you want to reach out and give us some more feedback. So hopefully everyone took notes in their discussion groups. Brad and I will go and revisit it. I know in our group we had really great ideas already from the participants, so we're gonna have to look into that. And yeah, again, if you do feel like you have things to contribute right now to I5K and want to join us, please reach out to us. You can find information here with this QR code. So feel free to head out. Appreciate everyone showing up for this longer 3-hour session for BG 2024. So this is a good way to end it. Thanks to all speakers if you're still there. And yeah, have a good rest of your day wherever you may be. Take care.
Thanks, everybody.
Thanks so much. Very cool. I'm
Bye-bye.
excited to get involved in any way I can. So maybe
Okay.
reach out to you, Monica.
Oh yeah, famous last words. We'll definitely reach out to you. Be careful.
Careful what you ask for, but in a good way.
Sounds good.
Indeed.
Yeah, great initiative. Awesome.
All right. Okay. I will stop sharing. Bye. And then we need Glenn before we stop. I want to make sure. Bye, Mark. Thanks for contributing. Bye, everyone.
Hi, Kevin.
Parents.